Our next contestant is Ellen Risemberg from Bioinformatics and Computational Biology with advisors Will Valdar and Martin Ferris. Ellen's project is entitled Genetic Susceptibility to Severe Coronavirus Disease. Ellen.
Okay. Animal to human transmitted coronaviruses have been a huge threat to human health over the last 20 years. SARS and MERS have infected tens of thousands of people globally, and COVID-19, of course, has infected many more and is at the forefront of our minds. At the same time, climate change and our increasing interactions with animals increases the likelihood of future coronavirus outbreaks. So it's important for us to understand how coronaviruses in general produce disease in humans so that we can better control those outbreaks. We can see with all of these coronaviruses, but especially with COVID-19, where outcomes range from asymptomatic to lethal, there's a ton of variation in how individuals respond to coronavirus infection. It's well understood that some of this variation can be explained by age and comorbidities, but my research is interested in understanding how our genetics may play a role. Specifically, I'm looking for genes or genetic variants that may impart susceptibility or resistance to severe coronavirus disease. We're doing this in mice. We use mice as a model organism because we share a lot of our genetics with mice. So the Barrick lab here at UNC did an experiment where they identified a coronavirus susceptible and coronavirus resistant mouse strain. They crossed those two strains with each other, and then the offspring of that cross has a genetic structure that allows us to identify genes that are statistically associated with that variation in susceptibility that we observe. So we infected their offspring with one of 3 different coronaviruses, the viruses that cause SARS and COVID-19, and then HKU3, which is a bat coronavirus, thought to be a candidate for future animal to human transmission. We observed their infection response and collected data on several traits, including weight loss, which we use as a proxy for disease severity. And then I can use that data combined with their genotype data to do a statistical analysis called quantitative trait loci mapping, or QTL mapping. And with QTL mapping, I'm basically looking for genes where the genotype at that gene is correlated with disease severity. And that indicates that the gene is influencing how the mice are responding to coronavirus infection. So in this way, I can look for disease-associated genes. I can do additional experiments and analyses to try to understand the mechanisms by which those genes are influencing disease outcome. And then this information, when applied to humans, can provide predictive biomarkers or potential drug targets. And since I'm studying 3 different coronaviruses, including one that's not yet in humans, I can hopefully identify mechanisms of genetic susceptibility that are shared across coronaviruses. And then this information can be used to help treat and control both current and future coronavirus outbreaks. Thank you.